Taking a look at Unit 3, Lecture 2, with the topic being infection. So here we're going to take a look at what are the characteristics of microbes that make them infectious, and what are some of the consequences and even mechanisms of infection. So starting off with the definition for a pathogen. Pathogen is essentially an organism who has a toxic, harmful relationship with the host. There are different types of relationships. Some of them are mutualistic, some of them are commensal, and then some of them are parasitic. And a pathogen is sort of another type of way of describing a parasitic relationship. It's important to note, as it says here, that this depends on the host, which means that sometimes the relationship could be non-toxic, even if you're talking about the same parasite and or pathogen. As a quick metaphor there, imagine you have three people, person A, B, and C. Person A and B can get along, however, person B and C have a toxic relationship. The relationship between person A and B is fine, B and C is harmful. Person B is the common denominator, and so only one relationship is toxic while the other is not. And that's kind of how microbes are. A microbe can essentially have a different kind of toxic relationship with one host but not the other. So when you look at one of the aspects of microbes that make them pathogenic, there are certain microbes who are just generally pathogenic across the spectrum, more in direct reference here to human beings. These are often referred to as true or primary pathogens, and we'll get into what exactly makes them fall into this category. There's another category that are referred to more commonly as opportunistic pathogens. So these are microorganisms who tend to cause disease given an opportunity, which is really in reference to one of two things. Opportunism relates to a weakened host immune response, being immunocompromised hosts or individuals, or a location in the body that naturally lacks immune cells. So these would be body locations that are naturally absent white blood cells or other immune protective factors. So the common denominator there is that opportunists take advantage when the body's immune system is either absent or weakened, and that's the common denominator. So sometimes you're unhealthy and that's why the immune system is compromised, or you just have normal areas where the where the immune system is not naturally robust. So the outer layer of the skin is a good example. The most outer layer doesn't have an immune response to it, and not until you get deeper into the skin tissue. And then also areas like the urinary tract is another example where the body actually relies more on the microbiome as a way to kind of keep unwanted microbes out. So that can also be compromised. So here's another list of factors that can weaken the defenses. So when you're looking at the specific aspect of having a weak immune system, you look at age when you're young and old, genetic defects that are required, such as AIDS, which is spread by the HIV or caused by the HIV virus. If you're a surgery transplant patient, you take immunosuppressant drugs, and that makes you immunosuppressed because you're trying to keep the body from rejecting the organ, for example. And another series of examples here as well. Now, when you get into what is it that makes something a true or primary pathogen, there's really a handful of things that go into this. The first is that we have to understand that there's a term called virulence. So virulence is your degree of pathogenicity. So if you're a primary or true pathogen, you have a high degree of virulence. And that's ultimately determined by, number one, your ability to establish, and then number two, your ability to cause damage. Establishment is actually the tricky part. Damage is the easy part. So when you often notice that you have an infection, you're noticing the damage. What you often don't notice is the fact that the microbes snuck in, established, did those prerequisites first, and then cause damage thereafter the fact. Okay? And so establishment is actually the tricky part here. Damage is kind of the easy part. Anything that contributes to virulence is considered a virulence factor. Relatively straightforward term there. We've talked about a capsule in previous lessons where the capsule makes it hard for bacteria to be phagocytized, and we'll come back to that here in this lesson as well. And the last note here is that, as I already kind of mentioned, establishment is a prerequisite to damage. So you have to go through the process of establishing before you can cause damage. And we'll look at establishment in more detail here on the next slide. So establishment, a prerequisite, is ultimately the setup for damage. So during establishment, for example, the microbe has to enter into the body. Then, in almost all cases, they have to attach somewhere to the host tissue. Then, in order to be there long enough to actually do damage, the microbe has to survive the body's host defenses, which would essentially be your immune system, more or less. So that's really what establishment is all about. And it's only really after the microbe has survived and not been thoroughly eliminated that it can then start to cause some type of damage. Every microbe has a different specific way of accomplishing these things. Some microbes can cause damage relatively quickly, but most of the time it takes several days and sometimes longer before you really start to notice the damage. But that is highly variable. So ultimately, primary pathogens have the greatest ability to establish, and that's a key aspect there that's worth understanding. The main difference between an opportunist and a primary pathogen is that primary pathogens have the greatest ability to establish. Opportunistic pathogens are those that can cause disease, which is the second part of virulency, but don't establish very well. And when we look at establishing, opportunists usually have a hard time surviving the host defenses. That's usually what it comes down to. It's not so much that they can't get in or attach, it's that they usually can't survive the host defenses. That's, again, it's sort of a general statement, not always exactly the case, but that's a general difference between the two. So, and just to clarify there, opportunists, if they get into the body in key locations or more commonly within a compromised immune system, they can end up causing just as much damage as a primary pathogen. So you can't really gauge the damage until based upon that, whether they're primary or opportunistic. The difference is how well do they establish in the host. So when you're looking at entry, a part of the establishment process, the body can be looked at through the context of portals of entry. So, for example, the skin, which is normally a barrier, could be compromised through cuts, nicks, abrasions, burns, things of that nature, needles, puncture wounds, insect bites, for example, and that then becomes a portal of entry. The gastrointestinal tract is essentially what you eat and ingest food and liquids through, and that then becomes a portal of entry, one of the more common ones. The most common is the respiratory tract, or constantly breathing air. The air can be full of pathogens, potentially. The air then becomes a portal of, the air becomes a temporary reservoir where microbes exist, usually transiently, and then the respiratory tract where we breathe them in becomes a portal of entry. More often, sexually transmitted infections, STIs or STDs as they used to be called. This is where many microbes enter through the urogenital tract. Not exclusively sexually transmitted, but very often that's where those are associated. Very often, microbes will enter through a characteristic route, so you can usually sort of say this particular pathogen comes through this particular route, such as an influenza, COVID-type virus, generally comes through the respiratory tract. Something like a fungus, in this particular example with athlete's foot, is going to take advantage of openings in the skin, usually other factors that go along with that, but through the cracks in the skin. And so if you were to, say, swab influenza onto your skin and not breathe any of that virus in through the respiratory tract, you would not get influenza. It needs to go through the respiratory tract. Vice versa, if you were to, say, swallow athlete's foot, you would not get it. It would not enter through the right portal of entry. So portal of entry is usually a very specific route. Now, there are some exceptions to that, and certain pathogens are more broadly adapted. So, for example, Staphylococcus aureus, this is one that's maybe referred to as MRSA. When it's antibiotic resistant, we call it MRSA. But Staphylococcus aureus, it can actually cause infection in almost any portal of entry. one of the most broadly adaptable pathogens out there. Tuberculosis, the bacteria that causes tuberculosis, Mycobacterium tuberculosis, is able to enter both through the respiratory and the gastrointestinal tract. So not every microbe has only one. They usually have one, and here's a couple exceptions to that. Now, as a microbe is entering through a portal of entry, the next thing that you want to recognize is that there's usually a minimum dose necessary to cause establishment. So here we're referring to what's called the size of the inoculum or the infectious dose, specifically. This can sometimes be abbreviated as the ID of a microbe. The ID is something that you can put specific numbers to, but it usually is more of just a broad concept. And the idea is that microbes with a smaller infectious dose are actually more virulent. So there's an inverse relationship. If it only takes a few cells to make you sick, that means that those cells are more potent. They can cause greater damage even in small numbers. So in contrast then, microbes with a large infectious dose are less virulent. So in this situation, you may have to swallow large amounts of foodborne pathogens, for example, before they make you sick. We might actually be exposed to foodborne pathogens in small doses pretty commonly, and you don't normally get sick. But if all of a sudden you eat undercooked food or food that was, say, prepared on a contaminated surface, and all of a sudden you're now swallowing millions of those cells, you're going to probably get sick because the body is sort of overwhelmed with the dose. So a certain number is required to cause infection, and if the dose is not sufficient, then the infection will not become established. Okay, and it's worth noting here, as it says in this last line, this relates back to the host condition. So if you're immunocompromised, you're more likely to have infection even with a smaller dose. So it's not. That's why it's hard. You'll see people put numbers to this, and you can do that, but it's not really as accurate to say that because it's just more of a relative thing. For example, if you have a compromised immune system, eating out at a restaurant can be dangerous to you because even small numbers of pathogens can make you sick, whereas most people aren't at risk because even if they are exposed to small numbers, their body will fight it off. They're not going to have a serious likelihood of infection. So immune compromised people are just generally speaking more at risk even with smaller numbers. Now, so when you're looking at establishment, microbes have to come through a portal of entry in a sufficient dose in reference to the host condition, and then they need to be able to attach in almost all situations. So attaching is a way to gain a more permanent foothold. And this is often specific, or this is specific to the individual pathogen at hand here. But for example, you need some kind of attachment that's specific. We've discussed some of these in the past. Some of these are highly specific, such as viruses, where you have this specific virus spike in the host cell receptor, the glycoprotein. Some of these are less specific, such as how bacteria might use things like Fimbriae or pili, which are fairly generic, to attach to any kind of general cell in the body that provides a favorable environment, like such as in the intestinal tract, for example. So something like a virus is highly specific. Something like a bacteria is generally less specific. This figure here shows some examples. So bacteria using Fimbriae have an easier time sticking to most of the host cells in the body. Bacteria using capsules is another example. This is how bacteria often stick to our teeth, for example. And then here we see virus attaching to a host cell, which is specific with the glycoprotein and the virus spike. So bacteria will use things like Fimbriae, pili, capsules, slime layers. Fungi and protozoa will have a variety of surface proteins. We've talked about all these in chapter 4. We didn't really talk about these here, so I'm being more vague. Viruses use their spikes, and things like parasitic worms, you see here a hookworm, will use things like suckers, hooks, or barbs in order to attach, almost like a mouth grabbing a hold of the host. That's actually a real picture. Now, so a bacteria or a pathogen in general has entered through a portal in a sufficient number and then gained a foothold through some attachment mechanism. If you're talking about viruses, or sorry, excuse me, if you're talking about bacteria, fungi, and protozoa in particular, this next stage is really crucial for their survival. This is a little different when it comes to things like viruses, and it's even a little more different when it comes to things like parasitic worms. So, especially bacteria, the most common, but also fungi, less common. In order for them to survive on the surface of the host cells, they're going to have to be able to survive the host defenses. And one of the first things that's going to show up are cells called phagocytes. So phagocytes are cells that just are described as cell eaters. That's basically what the term phagocyte translates to when you go back to the Latin origin of the term. It stands for eater of cells. And this is a broad term that describes a number of different types of white blood cells, which we'll get into in the next week's lecture more specifically. But you have this process here by which the body's white blood cells will recognize a foreign cell and then sort of morph the cell membrane around that bacterium and engulf it, and then enclose it in a special digestive membrane that will then essentially chemically digest the bacterial cell or the foreign cell inside of the white blood cell. And this is one of the first things that microbes are going to be exposed to. Again, this is mostly pertains to things like bacteria and fungi. They're going to encounter these cells once they enter, and if they can't survive the onslaught of this host defense, they're not going to establish. So that's why it usually requires that the host either has a few number of these white blood cells because they're immunocompromised for different reasons, or that the host is overwhelmed with a large number of cells, so that even if you have a healthy immune system, the body can't get rid of them fast enough, right? Or they get into a location where these types of cells don't exist. So that's one of the most common ways that the body attempts to remove these types of cells early on in the establishment process. Now, when you start looking at primary pathogens or true pathogens, one of the things that they can do is actually fight back, so to speak. And there's a class of mechanisms here that are referred to as antiphagocytic factors. So this is a sort of broad idea of a virulence factor that can essentially mount a defense against these phagocytes. So just a couple general strategies here. Number one is you avoid the phagocyte and you kind of play hide and seek or run away. The other is that you actually try to kill or inhibit the phagocyte outright. And you do this by sort of releasing some toxic substance that makes it difficult for the phagocyte to function. So that's when you start really looking at a more true pathogen, a primary pathogen. Even if the body can mount a defense, they have a defense against that. There's like a counter defense. So a couple of examples here on this slide. If you look at when you look at how to get away from a microbe, you can put up a barrier. Okay, running away is maybe a bad way to think about that. So when you look at avoiding, you can essentially put up a layer around yourself as the microbe to kind of keep that phagocyte from getting to you. So where bacteria often thrive in this is secreting slime layers or producing capsules. And these are both a type of extracellular secretion. And so when this happens for slightly different reasons, the phagocyte has a hard time actually ingesting the microbe, and the microbe becomes resistant to that process. Some examples are mentioned here. One of the bacteria that cause common pneumonia, Streptococcus pneumonia, Salmonella typhi, this is what causes typhus, typhoid fever, and Neisseria meningitis, which is a cause of bacterial meningitis. Now, less common and even more nefarious, these are like genuinely harmful pathogens. They can actually survive inside the phagocyte. So the phagocyte will swallow the microbe and successfully do that, but inside there has to be a process by which the bacteria is fully digested. So the digestion process is actually circumvented such that the microbe is engulfed but not destroyed. And it's almost like they can survive inside the stomach of the phagocyte. And so this ultimately is even worse because if the microbe gets away from this phagocyte, there are other immune factors that can come and clear it up later. So this is a first defense, but it's not the only defense. Why this is a problem is because the microbe can actually get into the phagocyte and then be sort of protected inside the very cells that are meant to kill it and remove it. And it's basically like hiding out behind enemy lines, and the body's other immune system processes can't see it there. And so it's actually stubbornly protected inside the very cells that are meant to protect it. Some examples here. This is how tuberculosis works, for example. And one of the more common examples, when you get tuberculosis, Mycobacterium tuberculosis can actually survive inside these cells and then evade the body's immune system. And this can take years. You can actually theoretically have this bacteria in your cells and never develop the symptoms, or it can play out over years and years and years and years. Some tick-borne diseases operate this way as well. Another common example of antiphagocytic factors, or a broad class of chemicals called leukocidins. These are substances that are released by bacteria that are toxic to white blood cells. So leuko is in reference to leukocytes, which is a way of describing white blood cells, and cidin is in reference to a poison or a toxin. So this describes a toxin to white blood cells. These toxins are broad, but the general outcome is that they lyse or disrupt the membrane of the white blood cell attempting to phagocytize. Now, once a microbe has made it through the establishment, they can now start to cause damage, given that they have the right genetic factors to damage the body. And we'll talk about some examples here. But so this part now takes place once the microbe has established. They've entered in the proper number, they've attached, and they've survived the host immune system, at least temporarily, not indefinitely, but usually the first wave of attack has been survived. And so now they're there long enough in sufficient number, they can start causing damage. So one of the first ways that you do this is often referred to as direct damage. And this is through the release of damaging enzymes and toxins. So the microbe has a genetic capacity to release enzymes or toxins that directly harm the host. This is often described as a direct damage to the host. A slightly less obvious way is often referred to as indirect damage, and this is inducing excessive inflammation. In the next chapter, we'll talk more about inflammation, but inflammation is the body's response to some kind of trauma. It doesn't necessarily have to be infection, but in this context, it would be infection. So when your body notices that you have infection, you start to respond with inflammation. Okay, again, we'll talk about more what inflammation is more specifically, but it's a process that is meant to get you aware that you're sick, sort of make you take different lifestyle precautions. You lay down, you sort of rest, and you let your body start to heal. And you also increase your fever and your temperature, and you start doing things in order to sort of change your behavior to make you more likely to survive. That process in the short term is good. However, and within the right extreme. So a little bit of inflammation for a short period of time is good. However, if you have lots of inflammation, and especially for a long period of time, that's bad. And certain pathogens have a way of getting the body to overdo the inflammatory process, such that the very part of the body's healing that's supposed to help you ends up actually hurting you. The other way that's been recognized more recently is an epigenetic mechanism in which the body's DNA, the cell's DNA, often near the site of infection, are altered in a way that can actually start to change the very host cells and their mechanism of functioning. This could change the way that they secrete their natural proteins, affecting things like hormones and nerve activity, or it could also affect the way the cells divide and replicate, which could actually lead to things like cancer. And you can actually see research that shows that intestinal pathogens have actually been correlated to increased risk of intestinal cancers, things like colorectal cancers and even pancreatic cancer to a certain extent, although that's more hypothetical. Um, and so stomach cancers, intestinal cancers, colon cancers, these are at least correlated with infections that are thought to maybe damage the DNA in those cells and ultimately disrupt them, leading to cancer. So that's a newer recognized mechanism there. And just to clarify something I said, point number 1 here, this is often referred to as direct damage. Point number 2, this is often referred to as indirect damage. And so inflammation is an indirect way of causing harm. Okay, so examples of enzymes and toxins, again, a mechanism of direct damage. You see here in this figure what that might look like. These here are the host cells. This is the nuclei. This is a sort of barrier around the cells that kind of glue them together. Here they call it cell cement. And as these bacteria in green release these enzymes here, these little specks, they can break down the cell cement in this context, damaging the cells and even spreading themselves from one part of the body to another. So in this process, they damage the body and also extract nutrients from the body tissue itself. So that's kind of what direct damage would look like in this context. So some examples to give you an idea of what those might look like. The enzyme mucinase is an enzyme that breaks down protective mucus. So mucinase or mucinase is an enzyme that directly breaks down a natural barrier, which is the mucus. Keratinase. Keratin is a natural protective barrier, very often found on the outer layers of the skin, part of the epidermis. And this is part of the way that the outer layer of the skin is protective. And here you have an enzyme that breaks that down. Collagen is similar. Collagen is a key building component in the body's skin. Collagenase is an enzyme that breaks that down. And hyaluronidase is an enzyme that breaks down hyaluronic acid. And that's actually what was being shown in that last slide there. The cell cement, here they call it cellular glue, and it often kind of holds cells together. So hyaluronidase, just to clarify here, is what you're looking at here. It's not labeled this way, but this is representing hyaluronidase, breaking down the hyaluronic acid here. So anyway, those are some of the many examples that you could cite here. These would be more involved in the body, in the bacteria pathogens progression from the outer layers of the skin through into the deeper dermal layers of the skin, for example. So those are examples of direct damage mechanisms. So in reference to indirect damage, inducing excessive inflammation, the body is adapted to keep the immune response relatively short and relatively contained. This is actually going to be chapter 15 now. This is updated in the newest version, chapter 15 here, by the way. So we'll talk more about inflammation in that chapter. But ultimately, the main takeaway here is that another way to cause damage indirectly is by causing the host itself to end up gearing up, ramping up inflammation to the point that it actually becomes harmful. So this is something that could be genetic, could be based upon your life and your overall health. If you're, for example, eating certain types of food like sugar, nobody wants to hear this, for example, myself included, but just so you understand, as a side note, eating lots of excess sugar is directly known to cause an increase in inflammation in the body. And that alone could make you more prone to indirect damage during infection. Your body is more likely to be inflamed depending on what kind of foods you're eating. That's also true for things like seed oils, which are unfortunately common in the Western diet. Seed oils are directly associated with an increase of inflammation. It's complex. I don't want to get into why that is here, but you could look that up if you're interested. Sugar and seed oils are directly associated with increased levels of inflammation in the body. So if two people get sick, genetically similar, two different diets and lifestyles, one person with a bad diet is more likely to have inflammation. So that's kind of the interplay between the microbe and the host, goes from genetics to your overall health, which is often a part of your overall diet. The example that's often cited here in the book is the pathogen Streptococcus pneumoniae, where the bacteria has a capsule, which is a direct mechanism of avoiding phagocytosis. The phagocyte has a harder time being eliminated, and over time, as it builds up, this causes inflammation as the body can't get rid of it, and then the inflammation ends up causing damage to the lung tissue over time. And this is often associated with secondary infection. So it's usually the fact that you have a virus and then the body's immune system is tied up. You can't engulf and phagocytize, so you don't have as easy of a way to get rid of them, so they persist longer, and then the combination of the virus plus the bacteria ends up causing increased inflammation over time, and that damages the lungs. Epigenetic changes are complex, and these are a newer mechanism that has been noted in the last 10 years or so. So still a lot of ongoing investigation here. But generally speaking, that the body's epigenetic mechanisms are like the switches that tend to turn on and off different genes. And so the general speculation is that bacteria in different ways can activate or deactivate this DNA. And when doing that, that could cause damage to the cell or loss of cell functions, generally speaking right here. And so as it says here, this is a recently discovered mechanism. So a lot of speculation, but more and more research sort of confirming that this is what's likely happening. Now, once microbes have gotten into the body, they've established and they're starting to cause damage, this next slide here looks at kind of where they might go at that point. So localized infection, relatively self-explanatory. It enters in one part of the body and remains in that part of the body. Systemic infection, I'm sure a lot of you are at least familiar with what that idea is. The body or the microbe enters in one part of the body and now spreads to different other parts of the body. And this is really in reference, by the way, to through the bloodstream is kind of the key part of that. It could be the lymphatic system as well, but another way you might just describe it is through the circulatory system. But usually it's described through the bloodstream. A focal infection is one where it's kind of like a systemic infection, but it's not traveling throughout the body. So you might have a microbe that enters in one area, travels to another, but it doesn't necessarily travel throughout the entire body. And so these are not common. This is more of an exception than the rule. But tuberculosis, for example, can enter through the gut and travel to the lungs. Strep throat, for example, could come from the back of the throat and end up affecting other parts of the body that might include something like the heart or the brain, for example. Toxemia is a term that describes sort of a different phenomena, kind of like systemic infection, but here what you have is a localized infection that releases toxins rather than the entire microbe traveling throughout the body. So in this situation, you wouldn't have the microbes spreading throughout the body, but the microbes spreading a toxin throughout the body. So it's sort of a variation of a systemic infection. And toxemia is something that you see, for example, in toxic shock syndrome, which is a staph-induced infection in the vaginal area, often with the ultra-absorbent tampons. And these are less common today because they've sort of changed some of the ways that these are produced. But when you have too many staph bacteria, which is somewhat induced by ultra-absorbent tampons, that can lead to the release of rare shock-causing toxins that actually are spreading from that site of concentrated staphylococcus. And it's not the staph that are spreading, but just their toxin. So you call that toxemia. Not common. The two most common here, just for record, are local and systemic. Focal and toxemia are more of an exception than the rule, but you do see those occasionally. Now, just as you have microbes that enter through characteristic portals, microbes will also vacate through characteristic portals. I'm not going to go through all those here, but that's sort of a general phenomenon. The persistence of microbes here is describing the way that microbes can go through the progression of establishment to damage. So what you'll find is that some microbes come in and cause damage very quickly. This is, for example, what you see with foodborne illness. One of the ways that you can establish whether you have foodborne illness is looking at the time frame. If you ate something questionable and you get sick within 8 to 12 hours, you associate that with foodborne illness because it comes on relatively quickly. Some things, however, will go through a period of latency. And so latency is a time where you have a delay. And this makes it harder to then associate your symptoms with an infection because it sometimes could be years later. Things like the herpes viruses, AIDS, HIV, rather, Epstein-Barr virus. These are all these are all infectious agents that can ultimately have a long enough period of time between exposure and symptoms that the exact cause often goes undetected for a period of time. And it's also possible for microbes to have flare-ups. And so you actually do see this with things like Epstein-Barr virus, also diseases like malaria and Chagas disease, both protozoa. They can actually have a symptom that pops up and then goes away and then comes back later and then goes away again. And so that can make it even more complicated. That actually can be useful in diagnosing because recurrent flare-ups are usually associated with those types of parasites. But the point is that latency is a part of the process that makes certain pathogens harder to identify. So this kind of relates to infections that go unnoticed, sometimes referred to as asymptomatic infections. These are infections ultimately where the person can be harboring the pathogen without showing any sign of it and at the same time transmitting it. So this can ultimately make it more likely for someone to be spreading the pathogen, given that they are not aware that they have it. This, for example, is common in the HPV virus called the human papillomavirus, in that males often don't show any signs or symptoms. However, they can be transmitting it, meaning that they're not taking any precautions, not changing any lifestyles or factors or anything like that. And so it's one of the things that is a contributing factor to HPV being one of the most common types of sexually transmitted diseases. Now, another concept in the spread of pathogens, so we just talked about the rate at which the symptoms come on. If your symptoms come on quickly, you're more likely to mitigate your spread. If they come on slowly or you're not aware of it at all, then you're less likely to change your behavior, more likely to spread it. Reservoirs are a different concept that sort of describe where these pathogens come from. So you have reservoirs that could happen naturally, the living and non-living world. So living parts of the living world would include things like other humans, animals, plants even. Non-living reservoirs could be things like the water, the soil, or the air. So a reservoir just describes where you're likely to pick these up. When you look at animals' living reservoir, a term comes up called a vector. And a vector is basically anything that can transmit an infectious agent from one host to another. Now, as it describes here, the term vector has different context depending on what you're talking about. So if you go back to the section on biotechnology, vector is a term that describes cloning, but here it's describing a pathogen, the transmitting agent of a pathogen. When you look at non-human animal vectors, because technically humans are an animal, so animals, not including humans, one of the most common types of vectors are insects. We all are familiar with mosquitoes, ticks. There's also things like biting flies, not as common. Sand flies are an example. Fleas are another example. Again, in today's world, not common, but fleas can transmit a variety of diseases. This is how the bubonic plague spread, the Black Death. Although we can treat it today with antibiotics, fleas can also transmit things like worms. This is how dogs and cats get a form of tapeworms, for example. Less common, but a concern are various types of mammals, such as bats, for example. Types of birds, a lot of concern about the bird flu, which can be spread by a variety of different types of birds. And reptiles can spread more commonly bacteria on their skin. Anytime you're handling some kind of reptile or amphibian, frogs, turtles, lizards, there's always at least some chance that you might get salmonella if you're not careful. Usually, it's one of the reasons why you always want to wash your hands really good, for example. Now, a vector then can occur in what's called a biological vector, and this is differentiated between a mechanical vector. So I'll go ahead and put this all up here. So basically, it's a differentiation between the two types here. So a biological vector is one that is part of the life cycle, and a mechanical vector is one that simply is spread usually on the outside of the skin, kind of like I just mentioned in the lizard there. So a lizard might have salmonella on its skin, and if you were to pick up and handle that lizard and then not wash your hands and then eat food directly after, you might contaminate yourself in this way, where the lizard would serve as a mechanical vector. The examples I gave here are houseflies, cockroaches. Houseflies are notorious for landing on animal feces and then landing on your food, and they can transmit bacteria and things like that in that mechanism. Anytime you have an insect that is part of the life cycle, however, we now have what's called a biological vector. So this is common with certain mosquito-borne diseases, where the mosquito doesn't just pick it up and transmit it on its wings. It actually has to go, the parasite actually has to go through the mosquito before it can then be transmitted to a different organism. So that's part of the biological life cycle, therefore we call it a biological vector. So, all right, we'll stop it there. And next week we are doing chapter 15, which is the part of the host defenses, where we look at things like the innate immune system and inflammation and that.